It's my pleasure to introduce Dr. Michael Lundie. Dr. Lundie is a staff cognitive scientist at ARA with deep expertise in neuropsychological research, cognitive modeling, and neuromodulation. His work spans brain health, cognitive warfare, and ideological polarization. Recent projects include evaluating tPBM to enhance cognition, neurostimulation for creativity, and tools to support TBI recovery in warfighters. Today, Michael will be sharing preliminary results from a sham-controlled study, and it's exploring how tPBM using the 1070 nanometer LEDs may enhance cognitive performance. So the presentation will run about 45 minutes, and then we'll have a Q&A at the end. So thank you so much for being here, Michael, and sharing your knowledge with us. So I stop sharing now if you would like to share your slides.
Well, thank you, everyone, for joining in today to listen in on this presentation. Very pleased and very honored to represent this joint effort by Applied Research Associates and University of Texas at Dallas, respectively represented by myself and Dr. Krosik. And so today we'll be discussing, hopefully we'll consider this a cleverly named little, not very well buried pun name for our study, illuminating cognition, augmenting executive functions through transcranial photobiomodulation. I think a natural place to start here is to go through some background work in this area. As we're about to see, this is some rather well-grazed territory in some respects by neuropsychologists, neuroscientists, and those who are trying to steer along the maturation of this technology and to see where we kind of fit into that trajectory, what contributions that we're hoping to have made on this front. So I think it's a good idea to not assume that everyone here is equally familiar with the basic science behind photobiomodulation and how it all works. So just a very, 10,000 foot above sea level view of how this technology works and how it interfaces with cellular physiology. The way this methodology interacts with our physiology is that near infrared wavelengths are highly absorbable by subunits within the mitochondria of our cells. And if we remember back to our high school biology classes, mitochondria are the powerhouses of our cells that drive metabolic activity and all the other sorts of physiological processes that cascade therefrom. The subunit is called cytochrome C oxidase. Once it makes contact with this particular wavelength range, of which 1070 nanometers is within that range, what results is an increase in the abundance of adenosine triphosphate, ATP, which is the energy unit that drives cellular activity. So you can imagine in the nascent stages of the exploration of this technology, physiologists were intrigued by this and wondering, okay, well, how could this perhaps be harnessed in some ways that are salutary to some of the desires that we have as human beings, where we try to invest in technology and interventions that might benefit ourselves with respect to cognitive performance, perhaps recovery from different disease processes. And so some of the background research in this area has been quite diverse. We look here in this pie chart to kind of look at the proportion of work and contributions along various pathways. You can see here some of it involving animal models and various brain disease states, sometimes just as an investigatory tool about the general mechanisms, cognition, etc. And I think there was actually a previous presentation that looked at on this front here that was hosted by Neuronic on long COVID recovery. All very exciting. And so, again, I know this is just a complete blizzard bombardment of text here. This is merely just to show the scope of work that's already been done on this front. Across all these different ways of operationalizing cognition, that is to say, using lab-based experiments to see how the mind works in action when presented with different kinds of challenges and problem sets, to kind of tease out the way our mind works to work through various types of problem sets and paradigms, to look at attention, working memory, memory recall, memory consolidation, etc. What we're looking at specifically would be the subset of processes that we refer to as executive functions, right? How as we kind of manage the resources of our minds in real time, dealing with complex problem solving type tasks. There was a study a few years back, 2022, by Zhao et al., and it was a science publication, so high caliber, high impact factor journal, looking at how laser-based tPBM over the right prefrontal cortex could be used to improve working memory capacity. That is to say, how the mental juggler, right, our ability to take in packets of information and organize that in our mind, and be able to perform sort of mental manipulations on those representations in our mind in real time, how that could be improved using tPBM. And they observed some significant improvements in their stimulation group compared to their sham group. And so that was intriguing to our group, and we wonder, since so much of what we do at ARA, we're a defense contractor, right? So just kind of letting y'all know why we really took an interest in this, wanted to sink our teeth into this work, was to see if there might be a more cost-effective means of replicating this finding using LED-based tPBM, but not just trying to replicate this working memory finding, but extend beyond that work and find other ways in which we can tease out and assess impacts of tPBM, let's say on sustained attention, that is to say vigilance, right? The kind of extending out the wick of our mind to be able to focus in on a task. And then also looking at other aspects of well-being, including stress reduction. So again, we already covered a bit of this, but just once again, so I was the lead on this particular project, which was an IRAD, Internal Research and Development Initiative within ARA. Dr. Krosik, who runs the reasoning lab at the University of Texas at Dallas, where he specializes in investigating the neural correlates, reasoning, judgment, decision-making. So as a team, we're very well equipped to take on this particular challenge. And so as we were assembling all of our resources and putting together this pilot study, we saw that the Neuradiant 1070 looked especially well suited to our efforts here, especially when you consider just the form factor itself, very convenient as a delivery mechanism for tPBM to our target that we had in mind, which is the prefrontal cortex. So those front two quadrants of the LED-based device you see here inside the chassis of the helmet, that's where the LEDs are arrayed, and that's how tPBM is delivered to the prefrontal cortex. So in our investigation, we didn't want to just look at the behavioral aspects, that is to say the performance aspects that we could detect using our tasks of choice, but we wanted to actually investigate the neurophysiology underlying some of these changes that we might be able to elicit. I had encountered a cognitive neuroscientist named Matthew Robeson, who at the time was at the University of Texas at Arlington, and he was using pupillometry as a way of interrogating the mind's ability to sustain attention over long periods of time. And basically, you can use the behavior or the mechanics of the pupils and how long one can sustain pupil dilation as an index of norepinephrine being released into the brain. We'll get into some of those mechanisms a little bit later on, but that seemed to be a especially well-suited neural correlate that we could use alongside the behavior measures for our investigation. And ultimately, it would be to establish the efficacy of tPBM to improve these key markers of psychological well-being and cognitive performance. So this is a pilot study, so it was relatively small sample size and scope, just to be able to pick up on a positive signal of a clear statistically detectable difference in performance between our experimental group undergoing tPBM and the sham group. So a total of 8 individuals, 4 in our tPBM stimulation group and then 4 in our sham group. And here you can see some of the demographics breakdown, skewed slightly towards a majority female. We had some exclusion criteria here that we wanted to ensure that we're working with healthy adults just to establish the impact of tPBM on that particular population before in our future work, we might look to more clinical populations. So you can see here, we wanted to ensure that any participant in our sample didn't have any outstanding neurological conditions, metal and skull, photosensitive medication, etc. And we also compensated our participants monetarily for each of their visits, and received IRB approval at the University of Texas at Dallas, naturally, since that was the site where we conducted our research and data collection. So for our methodology, We'll start here with the working memory task. So the way we set things up is that all participants went through 2 sessions, so within the same week. And in that initial session, they would either receive the stimulation condition, which would be 3 distinct rounds, 5 minutes each, of exposure to tPBM. I see that on the call, Will Dixon has joined us, and he was actually the individual I want to credit with giving us that recommendation, pointing out that the high throughput of energy into the brain with 15 minutes really redlines the circulatory plumbing, as it were. And so any sort of adverse conditions, which are relatively mild, like receiving headaches, could definitely interfere with performance. And so basically titrating with 5-minute intervals seemed to be a good way to kind of work around that as the brain adjusts to any of the acute neurophysiological changes that occur. And then those in the sham group, we would activate the device and then manually shut it off after a few seconds. And so that was what allowed us to implement a sham control type design. Another benefit, by the way, it turned out, is that it minimized the heat signature by doing 5-minute intervals. That sometimes can be a sort of tipping off of which condition one happens to be in. So that was another benefit of doing 5-minute intervals. And so within each of the sessions, participants would go through the a basic Qualtrics survey encompassing the depression, anxiety, and stress scale. Those are sort of index of well-being, and then a set of cognitive tasks, working memory, and then the brain gauge, and then psychomotor vigilance tasks. So again, starting with the working memory task, this was actually cribbed from the Zhao et al. Study. So what they did is they wanted to ensure that any sort of impact on working memory was not specific to the modality in which the working memory performance was being exhibited. That is to say, any sort of items you're having to put in the mental juggler, whether it's like, in this case, the orientation of objects on the screen, or matching the color display of different objects on the screen. They, the investigators in that study wanted to ensure, again, it wasn't modality specific. So there's a bit of that redundancy put in place just to be thorough in ensuring that tPBM actually did have an impact on the working memory construct per se, and not just one particular way in which that construct could be interrogated using a task. And in that study, they were able to show that in both cases that working memory performance improved. So we had 2 conditions nested within our working memory paradigm, a low load and a high load type of condition. That is to say, the number of items that a participant had to memorize, encode, and rehearse during a delay period that was on screen. So participants would see these objects in the orientation task presented to them. A delay period would occur for roughly 4,000 milliseconds, and then either the exact same screen would appear or a different screen, and the participant have to determine whether or not there was a change or not. And that's how accuracy was teased out and measured in this particular task. And then the color task, again, analogous, same thing. They go through a delay period, have to determine whether or not the same screen had been presented to them. And that's how we were able to measure working memory performance in our experimental design. And moving on to our brain gauge at the very top of the screen here, what you see here is that Cortical Metrics is the company that developed this device. It's very clever. Hopefully you all can see this clearly, but the device looks like a computer mouse. You know, I don't know if I'm embedded in the screen or not, but again, it looks just like a computer mouse, these 2 little prongs that are situated towards the very front of the device, and they vibrate. And the way it works is the cognitive task battery will go through different ways in which the somatosensory system can be measured. It was originally developed as a tool for measuring the severity of traumatic brain injuries. And so there's a process within the brain called lateral inhibition. What happens is that the brain, in order to maximize the sort of signal to noise ratio and sort of have like a fine-tuned representation of some sort of sensory event, it has to sort of quiet down all the rest of the cortical activities nearby. And so the brain gauge is this really interesting way of determining how well the brain is able to focus in on a stimulus of interest while shutting out any sort of distractions. And so, for instance, there would be one type of task embedded here within the design where the two prongs would vibrate simultaneously, but one would be slightly more intense than the other. And so the participant would have to click the prong that had a more intense magnitude of stimulation. And the idea is, could tPBM be used to augment the brain's capacity to make these very fine-tuned, minute determinations of one sensory state? And also, there's, besides accuracy, you can look at response time and see whether or not that is positively improved by tPBM. And so this would also be the first study of its type to use this device as a way of investigating the impacts of tPBM. So a way of kind of extending beyond the original study that inspired our design. And then lastly, we have the psychomotor vigilance task and pupillometry, which was overlaid during that task performance. So the way the psychomotor vigilance task works, it's relatively simple. There's a dot on the screen, and it's a little red dot that will appear periodically after a few seconds. And all the participant needs to do is click the space bar as quickly as they possibly can. And the speed with which someone responds is an indicator of how well focused they are. So if there's a sort of mind wandering or sort of drifting of focus, then there might be delays or like attentional blinks where the participant loses focus, and then there are these delays in response. And so the idea here is, could tPBM improve the focus, the vigilance of the participant so that they're able to remain task-oriented the whole time while taking this task? We use this Tobii eye tracker on top of the computer monitor that would key in on the pupils of the participant. And you can see here in this image, this example image, that the pupils, they constrict and dilate not just in response to changes in the ambient light. I mean, I think everyone in the audience is familiar with this. I mean, it's not surprising that our pupils will constrict and dilate based on changes in light conditions. But it also turns out that even if you hold light conditions constant, keep that the same, there are neurophysiological signals that will induce changes in pupil diameter, in this case, more pupil dilation, when we're in a state either of, say, stress or increased cognitive load or more demands on cognitive performance. And this is mediated by norepinephrine being released into the cortex. And so norepinephrine is a neurotransmitter that induces acute changes in our state of attention and focus. And so naturally, it occurred to us that mechanistically, this seemed to be a good candidate to cue in on to see whether or not changes in norepinephrine might be modulated by tPBM, and that in turn might be mediating the changes in behavioral performance, response time, etc., that we might be able to detect using the task itself. And so now we're going to go ahead and move on to our results. So very quickly, we for when it comes to the well-being question, right, just looking at aspects of depression, anxiety, and stress that might be impacted by tPBM. This is more of like the exploratory aspects of this work. You'll notice that we're not reporting any sort of inferential statistics here because the sample size is relatively small. But nevertheless, we did want to report some of this data in case it might be striking or of interest, and it turned out it really was. So what we observed, let's say for the depression category, for all these different categories, depression, anxiety, and stress, there's a scale, goes from normal, mild, moderate, and severe, and there's different point intervals that will separate the classification of an individual within each of those ranges. And what we saw for depression is that 3 out of our 4 participants in that stimulation group for tPBM had reduced their scores. The average reduction is about 3 points. For anxiety, everyone began with normal anxiety levels, but there still nevertheless was a score reduction, roughly of 6.5 points for the participants. And one participant actually decreased from 10 to 0, from session 1 to session 2, which was quite remarkable. Obviously still in the normal range, but still it's a quantitatively detectable difference. And then stress. All participants showed reduced stress levels, roughly of average of 6 points, and one decreased from 6 to 0, again with the normal range, but still something we wanted to remark upon and thought was noteworthy. And our sham control group, The overall trend was an increased score in at least one of the three dimensions, and relatively mixed results when it comes to depression and anxiety. Again, it's just to show that really wasn't much of a change, which is what you expect from a sham condition. So for stress, 3 participants showed no change. 1 participant actually showed an increase. So happy to elaborate a little bit upon that. Perhaps any clinicians here in our audience might have some thoughts on this. I would say this is not exactly in my wheelhouse, these particular constructs. I'm more so in the cognitive side of things, but I still wanted to report some of this for interest. It might be of interest to some in the audience who are interested in those aspects of tPBM. So for working memory, we were able to partially replicate the findings of the Zhao et al. Study, again, which used the laser-based tPBM. So in our case, what we showed is that in the high-load trials, that is to say when we have 8 items instead of 4 items that participants would have to memorize, and the composite score, that overall our tPBM group outperformed our sham group with an overall K-value score. By the way, K-values, again, just very quickly, this just is one particular way in which we can index and measure working memory performance. Roughly the way in which it's calculated is just the average number of items one is able to encode, rehearse, and then be able to properly deploy in a performance-based context within this task. And so higher scores are better, simply put. So like the scale, the max would be 8 because there's 8 items, right? That's the ceiling that someone could reach. So the closer you get to 8 items, the better the K-score is. And you can see the K-scores overall, looking at our y-axis here, overall were higher in our stimulation group compared to our sham group. Oh, one other thing I wanted to say. So for our color task, we did not observe this. I just want to make sure I make that clear. So it was only in the orientation task that we happened to observe this salutary effect of tPBM over sham. So kind of a curious finding that the color task, the performance levels were roughly similar for our sham and tPBM groups. For limitations of the study, we do discuss some of those considerations, but wanted to highlight that point really quickly. So for our brain gauge, there's a whole range of different constructs here that are assessed using this device. And of the different constructs that we tease out, there were 2 in particular that were highlighted as having a particularly remarkable performance difference across our 2 groups. And that was plasticity, so the capacity of the brain to sort of adapt to rule changes and complex task feature changes. Sort of the flexibility, the adaptability of the brain. This was more pronounced in our stimulation group compared to our sham group with a relatively large effect size. And then for reaction time, you'll notice it says reaction time 2. We had a higher performance in our tPBM group compared to sham. And the reason why it's labeled that way, and this is kind of getting to our conclusions, but reaction time 1, which is taken at the very beginning of this task battery, there was no real performance difference across our groups. But towards the end of the task battery, reaction time 2, that's when we start to see these performance differences. And so that leads us to infer that tPBM seems to be freeing up more metabolic resources, more cognitive reserve, to be able to sustain higher performance levels for a longer period of time compared to our sham group. And I think that interpretation is sort of countenanced by the fact that we do see this change specifically towards the end of the task battery. So again, there's a potential selective effect of tPBM on plasticity while stabilizing reaction time towards the end of this test battery. And then for our pupillometry, our psychomotor vigilance task performance changes, what we had observed here is that overall, if you're looking at the figure embedded within this slide, that we're plotting pupil dilation on the y-axis and the reaction time on the x-axis. And the smaller the number, the better it is when it comes to reaction time. Means you're faster. You're faster at being able to press that space bar when the cue appears on the screen. And then for pupil dilation, the larger the pupil diameter, this is an index of more norepinephrine being released into the brain. And what we see here within our comparison of the tPBM group in the turquoise color and the sham group in the sort of reddish orange color, that overall our tPBM group was faster. Overall, we're showing basically average scores across the 2 days that they received tPBM and larger pupil diameter. One thing we thought was interesting is we had one outlier in our tPBM group towards the upper right-hand corner of this graph, had the largest pupil diameter, but their relatively modest response time. So we this kind of gets to interpretation issues with pupillometry and what exactly norepinephrine is doing for the like how is it impacting performance. And we look at it as more so like a measure of effort, being able to put more effort into the task. And so one consideration here is that this individual was only ever in the tPBM group, and you have to wonder how this might have impacted their own baseline if, let's say, you were assessing them without ever having had tPBM. Perhaps they're able to produce more effort towards the task as a consequence of using tPBM, and norepinephrine is sort of an indicator of that. Again, this is just sort of interpretation issues, trying to understand mechanistically how is norepinephrine impacting performance. But in any case, it does seem to be mediating a change in one's state of attention or mitigating failures of attention during the task, indicated by stabilized or faster response times, which we also saw in the brain gauge. So just to kind of quickly summarize what our findings are here for working memory, one of our interpretations that we detail in a publication that we just had accepted in Military Medicine for these pilot study findings is that it's possible that the orientation and color tasks for the working memory modalities could engage different neural substrates. That's a possibility. So perhaps the neural pathways that are more so relevant to the orientation task are more dorsal, where we're actually where tPBM exposure is occurring, versus the more ventral pathways, which might be mediating the color recognition task. So again, this is just one possibility to explain why there might be a performance difference there. And then for our brain gauge, what we had inferred from our task performance here is that it seems that tPBM is stabilizing the availability of metabolic resources that mediate or interface with one's state of attention, being able to sustain one's focus and attention for a longer period of time without having to go through that refractory period, which would be more so associated with shifting into mind wandering or more default mode, network mode, where one is kind of drifting away from being connected to the task. And so that's the interpretation that we came up with for the brain gauge results, and frankly, even for the psychomotor vigilance task results, because in both cases, we saw an improvement and stabilization of response times in our stimulation group and not the sham group. So some of the limitations are, I mean, I would say that the fact that this is a pilot study, some of the limitations are pretty straightforward here and would be characteristic of the fact that we have a relatively small sample size. So for one thing to consider is that we did not take any baseline pupillary diameter assessments of our participants. So there might be some individual variability in our results that must be acknowledged. That is to say, whenever you see a pupillary diameter shift within the task, we don't know how much that deviates from whatever their baseline is outside of task performance. Right? So hopefully that makes sense, what it is I'm describing there as a limitation. And then one other thing to keep in mind about pupillary responses, pupillometry, pupillary diameter shifts are sort of causally overdetermined, right? Norepinephrine does impact pupillary change, but so does acetylcholine, another neurotransmitter system. So that has more so to do with memory performance. And so that's not exactly the most straightforward interpretation of the neurophysiology underlying the pupillary changes. And of course, small sample size, one has to keep in mind the statistical power is limited by that very fact. When it comes to design considerations, what I hope to do in the future is to do more crossover type designs. That is to say, a participant will go through a sham condition and then also shift into the stimulation condition to see whether or not we can compare someone's performance from, let's say, no stimulation, do the tasks, and then on a different visit, do the tPBM stimulation so that they become their own baseline. It just gets past the individual variability factor and increases statistical power when running inferential statistics on these performance parameters that we have assimilated. And there's also the question of the timing between the sessions. We had roughly 3 days on average of a gap period between stimulation period 1 and 2. And it is an outstanding exploratory question as far as what is the ideal time interval for having the most, let's say, if tPBM actually does have accumulating benefits with subsequent stimulation sessions, which does seem to be borne out by the extant literature, what exactly is that ideal time period interval to separate sessions? So that would be another limitation of our study. And here, just going to quickly tease out some of our next steps that we are going to embark upon, basically kind of bifurcated between military relevant directions and the more clinical applications. So for purely civilian clinical considerations here, our group is highly interested in neurodegenerative conditions such as Alzheimer's. We can see here that it's something that has caught the public interest for some time now, to see that Alzheimer's disease and various forms of dementia are going to continue to pose a serious public health issue, posing considerable costs and also impacts with respect to long-term care costs associated with an aging population. So it's definitely worth the investment and resources allocated towards this effort, using technology like tPBM to see if it can be used to stymie this trend. In fact, Maria DeCosta is also on this call, who has been a very helpful partner, and as we've been kind of working through this nascent research effort, is helping us with putting together a Alzheimer's intervention protocol. And so that's currently in the works and would represent an actual concrete step in that direction. So hopefully in the coming months, we'll be able to submit a proposal and see that come underway, and have yet another arrow in the quiver, as it were, for giving tools and resources to those within this population. And something close to me and my background is traumatic brain injuries. And so that was one of the first projects I worked on actually when I was a Ph.D. student in Dr. Krozik's lab years ago, focusing on interventions, cognitive-based interventions for rehabilitating the symptoms of TBI. And I think it's pretty straightforward to consider that TBI could be a viable target for tPBM, perhaps in conjunction with cognitive training. And here we can see that it's an issue that doesn't just plague the civilian population, but also military as well. And so this graph you see here is basically just showing, I know it's a little outdated, it's from a little while back, but it is a comprehensive snapshot of the number of individuals who have mild TBI or other forms of classification of traumatic brain injury, and perhaps the metabolic resourcing and vascular effects of tPBM would have a beneficial impact on the recovery process. And combine that with cognitive training might allow us to have a very targeted approach, a very systematic targeted approach to rehabilitating civilians and recovering warfighters who are struggling with this particular condition. And then there's also just the pure performance improvement capacities of tPBM. What you see here in the middle of this slide is a sort of trajectory or kind of a long game view of the maturation of tPBM technology. And we can see here that we're sort of in this phase of the in vitro, in vivo safety studies, where we're looking to see how exactly tPBM can be organized around operating principles, everything from, like, say, dose response, right? How much energy to put into the central nervous system? What is the ideal time interval for administering a given wavelength and intensity level to a particular cortical site to yield whatever performance shift we're looking to yield here, whether it's sustained attention, verbal fluency, working memory capacity, etc. So there's much work to be done on this front. And also, one particular variable that is especially interesting to me, because I have not myself actually put an experimental design together that actually looks at this differentiation between continuous wave, which is what we use in our study, and pulsed wave, say using the tPBM, where it oscillates at a frequency that matches that of a certain oscillation frequency, let's say for a heightened state of attention, or let's say for restfulness and sleep. In fact, there's a DARPA Restore program that's currently underway in soliciting abstracts and proposals to use a neuromodulation-based approach to improve the restorative capacities of sleep for the military under conditions of sleep deprivation. And so our team here is actually putting together a proposal towards that front, showing the great interest of the military in ways to improve the capacities of the warfighter. And so we're happy to be a part of those efforts. And here is just teasing out a couple of operational domains where this is especially important, where any sort of lapses in attention, attentional blinking, as it might be called, could be fatal. Say for pilots and special operators, it's possible that what tPBM could be used in conjunction with training protocols to augment the decision-making process and shorten learning curves to be able to basically facilitate maturation of expertise within various operational capacities and domains, and mitigating failures of attention through improved vigilance, through especially taxing conditions. So those are a few future directions that our group is interested in. And if anyone's interested in the references that were assimilated and presented throughout this presentation, feel free to reach out to me. I'm happy to hear any questions, comments, suggestions, any thoughts that anyone would like to share as we close in here this presentation. Very grateful for everyone's time and attention here. And it was a real pleasure having the opportunity to share some of my work with this group today.
Thank you, Michael. That was a fascinating overview of how you make the challenges and the changes in neurophysiology. And I see that we already have Patricia Jo Ryan has her hand raised. If you'd like to go first.
This is a very basic question, Michael. I apologize, but what does the T stand for? Because I'm...
Oh, yes. Transcranial. Transcranial, like meaning through the cranium.
But I wasn't sure, and I wanted to make sure that's what it was. Thank you. It was very exciting. I'm so excited about moves to take care of our warfighters and veterans.
Likewise.
Thank you. Oddly enough, I have twin cousins who are 82, who are both warfighters, one of whom is 82 and lives in Maine, just comes down here for a visit with his twin. And his twin really prevailed upon me to see what the helmet would do. So I did this in a very casual way. I didn't do this in a scientific fashion as I would normally do, because I really don't have testing materials, but I did finally get him to have a quantitative EEG. But I've been training him since the end of March, and he really did, I thought, have quite severe short-term memory functioning, and his Q indicated TBI. Well, he was Special Forces, jumped out of airplanes 40 times, so I would not I would expect something like that. And actually, I started very slow with just the programmed ones with glow and focus, and that was it. So I started with 10 minutes of glow and 3 minutes of focus, found that he had ringing in his head with the 10 minutes. So I backed off and went to 6. So we did that for a long period of time. And basically I've been training him probably anywhere between 3 and 5 sessions a week, just once a day. And this yesterday I queried him about if he's finding anything, because I haven't seen a whole lot of change in the short-term memory function. He said, I've noticed that his doing tasks, he is more aware of the sequence of the tasking, which I thought was at least something. And he is going to eventually, I think, continue with his daughter in Maine when he returns, because he's returning very soon. So this was my first opportunity to have a client, except myself, because I've been using it for 8 years, and to have a client to see. I was hoping for a little more, a little better functioning of his short-term memory than I got. But that was, I don't know, this is really so new to me, but I am so excited about what you're doing. And what is it about Texas that seems to be, they really seem to be further along than so many other states? Am I missing that, or is there other people see that? that you guys are really doing much more than, at least for the veterans and just for neurofeedback in general.
It's interesting. So I did my PhD work at the University of Texas at Dallas, as I mentioned, right?
Yeah.
We have a facility that is affiliated with UT Dallas called Center for Brain Health in the Brain Performance Institute. And it has been sort of like a pilgrimage site for different contributors within the space, whether we're talking about technologists who are trying to develop techniques like tPBM, and those who are clinicians and researchers all coming to collaborate. And it really has been an incredible hub Dallas is becoming. But I think, Brooke, you're in Austin, right? And I also, as far as I understand it, there's a lot going on there too. So for whatever reason, it seems that there's been a critical mass of those interested in this space to try to move the needle in the best ways possible. But to your anecdote, I think it goes to show that with any given construct that we're interested in impacting, whether it's short-term memory or any other construct, there's so many ways of operationalizing that, right? So there's ways in which an experimental task will try to sort of isolate it and put it in its it's like its purest manifestation, right? You're putting it in some kind of idealized laboratory type setting, and you might be able to see some changes there. But then when you go to more ecologically valid environments, what they're calling it, like where you're basically trying to see, does this transfer to, say, when I'm in the grocery store and I'm trying to remember my shopping list? When it comes to cognitive training, this has always been sort of like the litmus test, as it were, for determining the efficacy of any kind of approach. And what I like to see, and it takes a lot of clever experimental design formulation here, is to actually use those more ecologically valid tasks themselves as the index of the efficacy of the intervention, as opposed to using the laboratory type tests. So as an example, one thing we're trying to put together for some of our military applications is to find out, let's say for pilots, if they're doing some kind of flight simulator, perhaps there are ways in which we could index or measure performance within that task. That could be part of measuring the impacts of tPBM, because that's what one always hopes for, is that if I'm seeing some kind of change, let's say in a cognitive training or brain training game, whatever it might be, I'm hoping that's going to impact my daily life functioning. And I think that sometimes the cues that our brains use in order to navigate our perceptual space or whatever milieu we find ourselves in, that they're sometimes disconnected from the cues that we are becoming conditioned towards within a cognitive training paradigm. So the extent to which you can associate the performance with the cues in your daily life functioning, that might kind of speed along or produce more beneficial results for the impact of any sort of add-on intervention like tPBM. But of course, that's me just sort of speculating and kind of top of mind reflections on cognitive training and how it could be done in a way that's more beneficial to the end user.
Thank you. We have another question from Ginger Carney.
First, I just want to say something about a minor change I've noticed. I struggle with ADD, and maybe I'm having some progressive cognitive. When you have ADD, you have memory issues. So I don't know. But anyways, I've noted one of the things that I struggle with a lot is when I have to do authentication. I'm noticing that I remember the 6 digits much better. I don't have to go back. Usually I'd have to do it in 2 parts. And usually I'm very distracted because I'm doing something and then, damn it, I have to do this, task. And it throws me off in some ways. That's my one of the everyday life that I think is clear. Now, I also, I started this about the same time I started using the Muse headband. Are you familiar with that? The Muse headband, for people who aren't familiar, gives brainwave feedback. It's neurofeedback, and it gives brainwaves, a number of things. Mm-hmm. And then the new one gives oxygenation. I had used Mendy before, a little bit too, but I'm using these regular. So changes I have, it is a little difficult to tell. And then I've had an injury along the way and many distracting things. But the thing that seems most clear is the 6-digit. Now, I missed the very beginning, and you referred to the military a lot. So are you funded within military circles? Is your research done within a military funding source?
Yes. So as a cognitive scientist affiliated with Applied Research Associates, our company is primarily a defense contractor. So most of what we do is meant to translate to the needs of our DoD population that we service. That's right.
Well, and a lot of great things come out of military research.
Absolutely.
I'm wondering, how are you going to be affected by funding cuts? What's all going on otherwise? Will the military research of this nature also be cut?
I would say it's more so a reshuffling of things. Is probably what's going on here. Depending on whatever the priority changes are of any given administration, that's going to impact the overall vision for the research programs that are going to be rolled out by various departments and agencies. And so we as contractors try our best to kind of get a finger on the pulse to see how some of those trends might be changing. And so naturally, the kinds of research proposals we're putting together are going to reflect that attempt on our part to try to align as best we can with wherever the priorities happen to lie. So in this case, there's a great deal of focus more so on the performance aspects of the warfighters, trying to improve their ability to engage in their operational roles with as much fluency and effectiveness as possible, and less so on the rehabilitative side of things. So that's been a recent trend and change with the types of funding that is available for us to target to finance some of our research. So naturally, some of the proposals we're putting together are going to reflect that. So that's towards the end of the slide deck, as I was kind of teasing out the challenges that's faced by, let's say, by pilots and special operators. That flavor of research is definitely quite a bit of an appetite for that right now.
How about resiliency?
Resiliency is a tricky one, right? Because it's such a nebulous, capacious term, right? So some of that could be interpreted as rehabilitative, right? Someone's ability to bounce back from adversity or an adverse event. That's more so reactive or responsive to someone trying to rehabilitate from some sort of distressing event that has occurred at some point in their experience in their life. But there's also some aspects of that are preemptive, right? It's more so about building up the resilience of the system before you encounter an adverse event.
Right.
And so I would say that it's almost as though the interpretation of resilience, like of whoever we're partnering with, whatever, let's say, like any given government agency or any sort of research group we're working with, depending on the aspects of performance that we're trying to impact, that will impact the way in which resilience is actually construed. Like some aspects are very emotional, some of it have to do more so with like social connectedness and even intrinsic aspects of an individual, right? But of course, within the scope of our research here with tPBM, we're looking at more so the individual intrinsic capacities, let's say, of the nervous system, the integrity of the nervous system to be able to withstand adverse events. And one example of that would be sleep deprivation. One could infer or interpret that as like a form of resilience. So if I'm able to perform at a very high level, even with 3 hours of sleep, one could describe this individual as being resilient, at least in one way of construing that term. But some aspects of it are, well, I've gone through some sort of blast-induced injury that's impaired my brain. I'm already impaired, and now I'm looking to try to rebuild myself one piece at a time, already behind the 8 ball. And that's another form of resiliency, my ability to bounce back. So again, I guess I'm trying my best in a roundabout way to answer your question to show that it's a very complex term. And it's definitely not, there's no conventional universal understanding of what resiliency refers to.
Oh, and we didn't even used to use that term. That's a relatively new term. Now, I think it may be close to adaptogens, and I know at least in Russia, a lot of the research that was popular decades ago, I don't know if it still is, but is related to the development of things that increase adaptation, and some of the herbal adaptogens came out of that research. So, and I think that's very military relevant because you're dealing with adverse circumstances and how can you perform despite it.
And then there, you're kind of mapping resiliency onto, let's say, the interplay and the balancing of the sympathetic and the parasympathetic nervous system, right? When you bring into play terms like adaptogens and how you're able to kind of mediate between those two states. And if you're constantly in a state of highly stimulated sympathetic nervous system activation, you're really redlining the engine, as it were, in the brain. So it might facilitate the sustainability of that individual to engage in their performance long term in a healthy way if they're able to induce those shifts into the more parasympathetic state. Interestingly enough, the tPBM device that we use, the Neurodegenerative 1070, actually does facilitate those sorts of shifts. Like there's one that's like a peace mode, where I think the oscillation, the hertz of the LEDs is meant to entrain the nervous system to move to like basically like any sort of EEG readout you would have. You're shifting to a state that's more so like a restful mode, and that's one way to allow the brain, the nervous system, the physiology to sort of regenerate and restore itself. So, yeah.
Thank you.
Mm-hmm.
All right, West Center, Brain and Behavior Associates has a question or a comment.
Yeah, so I am a retired Marine and a combat vet, and I came into this field post-retirement. And I'm also here in North Texas. I'm in Fort Worth.
Oh, okay. Pretty much neighbors.
Say again?
Pretty much neighbors. Yeah, here in Dallas. So.
Well, yeah, as much as you can be a neighbor, an hour and a half away from everywhere in North Texas. Yeah,
that's right.
We've been using photobiomodulation here in our clinic in combination with mild hyperbaric oxygen. We have a 4-place chamber here, and we have the ability to do not only the seated, but we can lay people down, put red lights over the carotid so that we're kind of supercharging things on the way up, and also getting a little lymphatic drainage in the way. I will say to the person who made the question or statement earlier about resilience being new, we're doing resilience. When I first came into the military in 1979, with the post-Vietnam work that was being done by Dan Siegel and others, looking at could we predict who was going to get PTSD and who would not, based on extant resilience factors like education, socioeconomic, status and things like that were factor loaded. You can read those in the National Vietnam Veterans Readjustment Survey. It's a nice little summary of all that work that was commissioned by the Congress in the mid to early 80s. And so resilience isn't new, and at least in terms of the military. We've been doing it for decades. But we still haven't been able to break the code on psychological resilience as it relates to task performance in combat, with a variable that seems to be most important is, what kind of training did the person get? So that's why you tend to see higher levels of resilience in Rangers, Marines, and SEALs than you do the average line soldier. And it has to do with exposure to stressors that build up over time resilience. It doesn't mean those folks don't get PTSD. I've got a clinic full of them. They do. They just tend to get it at lower rates, even though they have greater exposure. What made you decide to use Brain Gauge? We use Brain Gauge here too, so it's a great tool. But what led you to pull that one out of the hat?
Yeah, so we are fortunate in that we have a strong track record of collaboration with Cortical Metrics on previous projects, focusing on cognitive performance enhancement, and I've been impressed with the reliability, the validity of the device. And it occurred to me that a great deal of performance assessment in this space tends to be mediated strictly through a limited number of sense modalities. Mm-hmm. I thought that approaching this in a multi-pronged way where we're interrogating the performance of the nervous system through somatosensory means, would also be an interesting way of putting a check on any sort of performance increases that we're detecting. Let's say with our psychomotor vigilance task, where this is mediated primarily through the visual modality. If we could see a response time shift that could be detected purely through the somatosensory channel, and you're seeing that also in the visual motor response channel, you're seeing a more of a domain general trend and the performance benefits of tPBM. That was one of the main justifications that we had for including the brain gauge. But it was also to be more clear and precise on the specific constructs, the selective effects of tPBM on cognition, because there is a broad range of different construct categories that the brain gauge will tease out. And so that would help us to sort of find demarcation points, clear delineations on the specific constructs that are impacted by tPBM in a more exploratory way, as opposed to us starting off by dint of an experimental design saying, well, we're just looking at working memory and we're looking at sustained attention and a response time. But to see, are there any other constructs, based on the versatility of the brain gauge testing battery, that we might have overlooked in an exploratory way, to be able to include that in our pilot study findings? So that would give us a positive signal to whether or not there are other potential pathways of development or applications for tPBM that might be available to us for further development.
So with vets, did you use TBI as an exclusion criteria, or did you by necessity have to include TBI in your inclusion criteria for your subjects?
For purposes of this study, this was strictly on a healthy adult population, which is 18 to 50. And so one of our exclusion criteria was having a history of a traumatic brain injury, any sort of neuropsychiatric condition that had been diagnosed, to ensure that we're able to keep this sample strictly within that range of the healthy population.
Yeah, good. Thanks for that. Appreciate you. Thank you.
Certainly. Thank you.
Michael, we've come to time, but there's one question in the chat, and Patricia has another question. Would you like me to collect those questions and email them to you, or do you have time to
I have time. I'm more happy to.
Okay.
I'm going to have to use my helmet because I forgot what the heck I put that chat on there for. So I'm finding that at my age, it takes me about 3 seconds to recover some of the things that I want to recall. So it's time for me to do a little more helmet. So sorry about that.
It's okay. No. Yeah. Any more questions? Happy to receive them. If anyone wants to reach out to me, continue correspondence, definitely always welcoming that as well.
Well, I can give you this one question from Subrat in the chat. Given that working memory capacity is supported by coordinated activity across the frontoparietal networks, like the DLPFC and superior parietal lobule, etc., have you explored or considered using source localized EEG or MEG to assess how tPBM modulates large-scale cortical connectivity?
Definitely. So one of the interesting prospects for future research on this front is to see whether or not there are cascading effects from our tPBM stimulation protocol. So again, we are targeting in a very broad way the prefrontal cortex, but of course there's a large symphony with various players within the frontal parietal network that mediate working memory and other aspects of executive functions. And it would be a good idea to see whether downstream there is any sort of change in activity or connectivity, perhaps coherence changes across these different nodes within the network. I think that would be a direction of future research that would be highly beneficial. In fact, for our sleep study, we're looking to integrate EEG as one of the data streams that we'll be collecting. And of course, that's very specific to that particular domain of trying to induce more of a restful state. And so the EEG oscillation signatures that will be interrogating are more so within that domain. But yes, I think that would be a part of the maturation of our understanding of tPBM and its neurophysiological impacts.
All right. Well, I think we should end it here. We've gone over a few minutes. But thank you again, Michael. It was really wonderful to see the potential for supporting cognitive function. Exciting to get a preview of the studies that you have in the pipeline. And we just have one announcement that June 5th, Dr. Andrea Arujo will present on tPBM for mild traumatic brain injury. And it's going to be a case study on brain function and psychometric outcomes. But thank you, Michael. We really appreciate it.
Oh, it's my pleasure. Thank you, everybody.
Thank you, Michael.
Thank you, Michael. Excellent presentation. We'll be talking soon.
Looking forward to it. Take care, y'all.